Assalamualaikum and good day everyone. As promised, this is the continuation of transcription factors and transcriptional control, which is part 2. These 2 weeks is about all of them, okay, on transcription factors and transcriptional control. But second part is actually a bit lesser or not as heavy as the first part because we have learned the heavy part, okay. So I'll lighten your burden, okay, in the second part. So let's move on to the regulation of transcription, okay, by the polymerase 1 and 3. Why not 2? Okay, let's take a look because it's the most important. So we'll have a look at the most important last because, well, you would guess it, okay, there's a lot of things, okay, to look at. So first and foremost, both RNA polymerase 1 and 3 transcribe limited sets of genes, which we know. You can refer to your previous notes. So we know that RNA polymerase 1 transcribe ribosomal RNA, RNA polymerase 3 transcribe 5S rRNA, tRNA, 7S lRNA, and U6, okay, snRNA. So both of them dominate cellular transcription, about more than 80% of total RNA synthesis in cells, and this is also required for transcription and also translation. Now, what are the promoters associated with RNA polymerase 1? So usually, okay, the promoters have to exhibit stringent species specificity, which means that for every species, okay, even from the same family, okay, they are very specific. Okay? Probably you will not find 100% similarities or homology. So little sequence similarities from different species, but functional promoters is of course highly conserved from yeast to human. Okay? So the keyword is highly conserved, but not 100%. Okay? So copies of rRNA genes, okay, are usually found as repeated clusters. So the promoter are housed in IGS or intergenic spacer, and the core promoter, okay, which is about 50 base pair upstream of the initiation site, is of course necessary for the initiation of the basic transcription. So it contains the ribosomal initiator or rINR, which is a conserved rRNA promoter sequence element. Now, you can basically see, okay, in figure 1, and there is also a description on what is what, okay, so you can ponder on it, okay, so you can look at your notes better. So because I would just summarize again from the figure. So basically, okay, ribosomal initiator contains a T-rich data-like sequence, but not an interaction site for the TBP. So remember your TBP, okay, if you don't refer back, okay, to your notes before this. Now, for the assembly and further stabilization of the complex on the copromoter usually requires other factors and elements, which is of course very obvious, isn't it? So the key element, such as upstream promoter element, okay, which is situated around 150 to 200 base pair upstream of the +1, is needed, okay? But it is not necessarily required for transcription initiation. It only just stimulates transcription by the core promoter. So we also have, of course, okay, initiator, okay, or basically a promoter upstream, okay, but we also need a terminator. So we have a PT, which is a proximal terminator, which, well, even though the name is terminator, okay, it actually stimulates transcription upstream of UP. And also it has some other functions, such as to protect promoter from wandering polymerases, remodeling of chromatin over the promoter, and architectural holding of the RNA repeats in nucleolus. So that's about RNA polymerase 1. So we move on to RNA polymerase 3, okay, the promoters. So we have a few types, okay, of promoters, okay. Type 1 promoter is for 5S RNA because we know RNA polymerase transcribe, okay, few types of genes or few types of RNA. So type 1 promoter is requires sequence element downstream of +1, but it should be within the transcribed region. So it is a discontinuous essential blocks of A and C separated by non-essential region somewhere in between them. So they are intolerant to changes in spacing between individual elements. So basically, this is of course from a journal as well. So further, elaboration is actually stated here. So you can have a look, okay, at your notes to read down about it. Now, second type of promoter, or type 2, okay, is for transcription of tRNA genes. So it comprises of highly conserved block A and B, okay. So the previous one is block A and C, so this time is block A and B within the transcribed region. So block A is homologous to block A of type 1, but much closer to +1, okay, so the location. Block B variable location. So it could be nearer to A block or could be farther apart, okay, to the A block. So again, okay, explanation is over here, so you can have a look at it. Now, third type of promoter, okay, is only available to minority of vertebrates lacking requirement for intergenic promoter element. So they have similar arrangement as in RNA polymerase 1 and also 2, okay, which is we have TSC, PSC, and basically upstream of the Tata box. Now, basically, okay, transcription complex assembly on RNA polymerase 1 promoter, okay, is as such. So it requires few transcription factors, so such as about one or two factors and single co-promoter binding factor. So the function of the co-promoter is to mediate transcription with the help of UPE, enhanced elements, and the PT. Okay, sometimes, not all the time. So co-promoter comprised of TBP and 3 to 4 additional subunits such as polymerase 1 specific TBP associated factors, okay, which is TAF. So TAF in turn functions, okay, basically have a similar function to TF2D and TF2B in RNA polymerase 2 and 3, which is to recruit your RNA polymerase and remain bound to the promoter to make sure that initiation of transcription could be initiated. So you have to also bear in mind, okay, TBP is used differently, okay, from RNA polymerase 2 and 3 in RNA polymerase 1 because it is not required for some in vitro studies. Well, I'm not going to elaborate further on this because it actually requires some other mechanism which is understood in certain organisms, which is mostly not understood in a lot of other organisms, so we will not delve too much into that. Now, furthermore, UPE usually binds to transcription factor, which function to aid assembly of cofactor to the promoter, but UPE is also used, okay, depending on the species as cofactor alone, okay, and it could also mediate basal transcription. And we have an enhancer just now, okay, so enhancer of course stimulate transcription, but the precise mechanism in RNA polymerase 1 is actually unknown. Okay, it is only known to act as a loading agent for promoter by supplying some limiting component. That's it. Now, we know that RNA polymerase 1 is recruited via protein-protein interaction with a cofactor. Well, basically, it's the same for the other types of RNA polymerase as well. So, polymerase can contact any DNA sequence. Hence, okay, initiation is actually at random. Okay, of course, okay, it could also occur in the absence of the cofactor. Okay, but not all RNA polymerase. So, this is in RNA polymerase 1. So, if the cofactor is present, RNA polymerase 1 will bind specifically to promoter and suppress non-specific initiation. So that means that, okay, transcription is very specific. Sequence alterations disrupting cofactor binding could eliminate RNA polymerase 1 recruitment. Now, let's move on, okay, to RNA polymerase 3, okay, how transcription complex, okay, is assembled. So basically, we know that there are three types of promoter. So type 1 and type 2 have certain similarities. Okay, they have two blocks. Okay, one is block A and C, one is block A and B. So TF3C recognizes block A and B sequences and recruit TF3B. TF3B contains TBP, which is of course, okay, we know, and can bind to the Tata box independently, but type 1 and type 3 promoters lack Tata sequence, hence it could not be recognized directly by the TF3B. So again, okay, if you just couldn't remember, okay, of the factors, TF3B recruited by protein-protein interaction, okay, usually contact DNA bound TF3C. So once TF3B, okay, is recruited, polymerase 3 is of course recruited as well, okay, to the promoter and position over initiation region, okay, to perform basically to form the transcription initiation complex. Now, for type 3, okay, so there are distinct factors, okay, that is required. 2TF3C is required, but mostly it only utilizes TF3C1, not TF3C2. Why? Well, it's a bit lengthy, so we will not go there. So PSE is being recognized by factor known as PTF or SNAP-C. So both PTF SNAP-C can interact with the Tata-bound TBP or TF3B, and PSE binding to the Tata box, okay, are usually slow. I wouldn't say very slow, but it's kind of slow rather than the other types of promoter. So initiation complex for U6 genes usually takes longer than tRNA or 5S RNA. So PSC occupancy is the most important in assembly and initiation complex, okay? Because basically then PTF and SNAP-C and TVP will enhance each other's recruitment to the U6 promoter. Now, how about transcript initiation and elongation? And this is, of course, we know occurs for all types of RNA polymerases. So initiation is, of course, we know it starts with the melting of double-stranded DNA, which allows access to template strand, because without the melting, without the double-stranded being melted or basically unwound, then you couldn't start off transcription. So in RNA polymerase 1 and 3, so the bubble forms spontaneously, and of course this is temperature dependent upon binding to polymerase. So the melted bubbles initiates upstream of the plus 1. For RNA polymerase 1, so it melts around 9 to 10 base pair before initiation. So initiation of the promoter is based on the clearance and followed by expansion of transcription bubble to around 19 base pair. It's actually not that accurate, but a lot of textbook and even some journals basically put in 19 base pair. For the RNA polymerase 3, it requires TF3B to melt DNA helix. So the bubble of the melted DNA moves as polymerase 3 progresses into the gene. So for the elongation, for all of them, it is basically kind of a dedicated elongation factors for RNA polymerase 1 and contains TF2S-like function of C1L subunit. So it recognizes pause slides and class 3 genes which are extremely short, so no elongation factor is needed. Now, for polymerase 1, gene terminators for termination is within the first 100 base pair downstreams of 28 as rRNA coding sequence. So the terminator is of course mediated by DNA binding protein. For RNA polymerase 3, the terminator is intrinsically recognized by polymerase. So it will recognize termination sites accurately and efficiently even without any factors. So this is one of the example being given by a study. So cluster of 4 or more T residues can serve as a terminator signals as compared to a string of A in polymerase 2. Now, how about reinitiation? So that means that probably it would have halted or stopped or completed, but you want to reinitiate it back. Can you do that? In certain cases you can, but of course there are some things that you need to bear in mind, such as for our RNA genes, so it usually have an efficient reinitiation to produce high density RNA polymerase 1. How is it done? So fully functional core factor left on the promoter usually allows for repetitive polymerase recruitment. This is basically based on theory that bundles of arranged rRNA in nucleolus causes two things. First, all promoters and terminators to be pulled together in the central core, and it later on transcribe rRNA and IGS looping up. So such arrangement will result in high concentration of RNA polymerase in promoter vicinity. Hence, reinitiation could be initiated, and you could have actually frequent reinitiation without any problem. For tRNA, and of course, we're talking about polymerase 3. So in yeast, stable complex can direct subsequent cycles more rapidly. So usually after first transcript, tRNA can be synthesized in lesser time, 35 seconds compared to 5 minutes for the first transcript. So how is this done? RNA polymerase 3 is recycled without being released from the template. So this is basically to avoid slow initial recruitment because it will take a long time. So they just basically recycle it. So this is there is also similar evidence in human done, but I actually couldn't find the paper. So to sum up, on the transcription factors and also the transcription control, we know that transcription is the key control point for gene expression, and we're talking about gene, not protein. For protein it's of course translation. So transcription factor proteins play a key role in regulating transcription, and we know that there are lots of transcription factor proteins. So there are two major categories of transcription factors. First, general transcription factors, GTF, which are a group of helper proteins which is of course needed by eukaryotic RNA polymerases in order to bind to any promoter regardless of RNA polymerase 1, 2 or 3. Second, there are specific transcription factors, which is of course okay in contrast or the opposite again of general transcription factors. Okay, usually it regulates the transcription of specific sets of genes. So this kind of specific transcription factors okay, typically act by binding to DNA sequences and helping or blocking the formation of the transcription initiation complex. For example, RNA polymerase plus general transcription factors at a nearby change promoter, and you have basically just seen it okay in the first few slides okay just now of TF3B okay, and so forth. So regulation of specific transcription factors is often combinatorial. So with a certain set of transcription factors serving as a key to drive transcription. But overall, okay, can you see that actually the recruitment or the order or the sequence of how the transcription factors, okay, whether general or specific, okay, when they come to form the loop is actually more or less similar for either RNA polymerase 1, 2, and 3. So this actually allows information from different sources to be integrated into a single response. Hence, the level of transcription of the gene could be controlled. Alright, so that will be the end of it. So, see you again.